Welcome to G Fun Facts Online. Imagine surviving the strike of one of the world's most venomous snakes. You endure the initial blinding agony. The fangs have pierced your skin, and in a fraction of a second, a highly complex cocktail of enzymatic and non-enzymatic proteins has been injected deep into your muscle tissue. You survive the terrifying, frantic rush to the hospital. You survive the systemic poisoning thanks to rapid medical intervention. But then, you wake up to a horrific new reality, only to face a lifelong disability. For hundreds of thousands of people across the globe, surviving the venom is only the very first battle. The subsequent war to save the affected limb is a war that is almost always lost. Snakebite envenoming is a profound and wildly neglected global health crisis that claims over 100,000 lives annually. Yet, even for the millions who survive, the aftermath is frequently devastating. Victims are routinely left with permanent disabilities, severe muscle destruction, and the complete loss of limbs due to the catastrophic swelling and localized tissue death triggered by the venom. Let us take a deep dive into the Asian Chinese moccasin, specifically the species Denecystrodon acutus. This venom doesn't just poison you in the traditional sense, it actively digests your tissue. It is packed with destructive enzymes that literally break down cellular walls and blood vessels. When these foreign toxins flood into your tissue, your body's immune system detects the massive incursion. It panics. It rushes specialized immune cells to the site in a frantic attempt to neutralize the threat. But in its absolute desperation, the immune system drastically overreacts. It triggers what we call an inflammatory cascade. This results in runaway inflammation, massive fluid accumulation, and ultimately necrosis, which is the complete, irreversible death of muscle tissue. The victim's own defense mechanism is hijacked, turning the bite site into a ground zero of cellular destruction. For decades, traditional antivenoms have been the absolute gold standard of care. They are a crucial, miraculous, life-saving intervention. They consist of antibodies that are painstakingly harvested from hyperimmunized animals. When injected into a human victim, these antibodies circulate in the bloodstream, bind to the venom proteins, and clear them from the body, preventing systemic organ failure and death. But they possess a glaring, tragic blind spot. While they effectively neutralize the circulating toxins in your blood, they are notoriously poor at reversing the localized inflammatory cascades and the severe tissue destruction that unfold rapidly right at the site of the bite. Ornella Rosetto, a brilliant neurobiologist at the University of Padua, points out exactly why this is a problem. Traditional antivenoms cannot reverse local damage once the fuse has been lit. Once that inflammatory cascade begins, the antivenom cannot stop the extensive muscle death. Furthermore, venoms vary wildly. They differ not just between different snake species, but even between snakes of the same species living in different geographical regions. Because of this, antivenoms are highly species-specific. Administering the wrong antivenom yields little to no benefit, and even if you get the exact right antivenom perfectly matched to the snake that bit you, it still cannot undo the localized tissue melting that occurs in the minutes and hours following the bite. The critical need for a universal, locally acting treatment led a team of researchers in China to an incredibly unlikely candidate. In a fascinating twist of modern medical science, they proposed an unorthodox solution, fighting fire with fire, or more accurately, fighting venom with another deadly toxin. They looked at botulinum toxin. You probably know it globally as the cosmetic wrinkle eraser Botox. But botulinum toxin, produced by the bacterium Clostridium botulinum, is arguably the deadliest natural chemical compound known to humanity. Think about that for a second. The most lethal natural poison on Earth. In its raw, unadulterated form, it works by cleaving specific proteins at the presynaptic terminals of your neuromuscular junctions. It permanently inhibits the release of a vital neurotransmitter called acetylcholine, severing the communication between your nerves and your muscles, causing severe flaccid paralysis and ultimately death. Yet, despite its incredibly lethal evolutionary origins, modern science has tamed this beast. Through precision engineering and extreme dose attenuation, scientists have transformed this microscopic assassin into a multi-billion-dollar medical and aesthetic powerhouse. Beyond just flattening out facial lines in beauty clinics, botulinum toxin has an extensively established track record in neurology. It is used to treat chronic migraines, severe muscle spasticity, overactive bladders, and cervical dystonia. But how do we get from paralyzing muscles to stopping snake venom? In recent years, neuropharmacologists started noticing that botulinum toxin possesses secondary properties that extend far beyond simple muscle relaxation. It turns out this deadly bacterial compound is a profoundly potent analgesic, meaning it stops pain, and an incredibly powerful anti-inflammatory agent. Studies revealed that the toxin's specific mechanism of inhibiting synaptic vesicle fusion doesn't just block acetylcholine. It also prevents the release of key pain-modulating and pro-inflammatory neurotransmitters. We are talking about critical chemical messengers, like glutamate, substance P, and calcitonin gene-related peptide. By forcefully halting the release of these specific messengers, the toxin effectively prevents plasma extravasation. That is the severe leaking of fluid from your blood vessels into the surrounding tissue. It dampens down localized inflammatory flare-ups almost instantly. This entirely unique mechanism of action made it a prime, albeit highly unconventional, candidate for testing against the rapid, localized destruction characteristic of viper bites. This brings us to a groundbreaking, truly revolutionary study published in the February 2026 issue of the journal Toxicon. A brilliant research team led by medical toxicologist Pin Lan at Lishui Central Hospital in China decided to put this wild theory to the test. Could the extraordinarily potent anti-inflammatory properties of botulinum toxin actually be weaponized against the muscle-destroying venom of the Chinese moccasin? Operating under strict, highly controlled laboratory conditions, the team injected rabbits with the raw viper venom. They carefully divided these subjects into 3 distinct groups to observe the effects. One group received only the venom, representing a standard, untreated snakebite. Another group received the venom, but alongside it, a precisely targeted dose of botulinum toxin. And finally, a control group was given a completely harmless saline injection for baseline comparison. Over the next 24 critical hours, the researchers closely and meticulously monitored the injection sites. They weren't just looking at the surface. They were actively analyzing the physiological changes, tracking the presence and movement of specific immune cells, and examining the structural integrity of the muscle tissue at a microscopic level. The differences between the groups were absolutely staggering. In the venom-only group, the results were as gruesome as you would expect. The rabbits suffered catastrophic localized trauma. Their affected thigh muscles swelled to more than 30% larger than their original circumference. When the researchers performed biopsies, the tissue samples revealed severe, irreversible muscle death and extensive cellular destruction. The venom and the resulting immune storm had literally melted the muscle fibers. But then, they looked at the second group. In sharp, unbelievable contrast, the rabbits that received the botulinum toxin alongside the venom experienced almost no swelling whatsoever. Let me repeat that. Almost zero swelling from one of the most destructive vipers on the planet. Furthermore, the extent of muscle tissue death was dramatically, remarkably reduced. The physical integrity of the limb was completely preserved. It was as if the venom's most destructive properties had been simply turned off. To unravel the profound mystery of how a paralyzing neurotoxin could perform such a miraculous rescue of living tissue, the researchers had to look deep at the cellular level. They specifically focused their attention on large, specialized immune cells known as macrophages. Now, macrophages are essentially the frontline soldiers of your immune system. They generally operate in 2 very distinct modes, which scientists classify as M1 and M2. M1 macrophages are aggressive, destructive, and fiercely pro-inflammatory. Their primary job is to attack foreign invaders, like bacteria or venom proteins, and clear out cellular debris by producing localized, intense inflammation. They are the scorched earth infantry. M2 macrophages, conversely, are the body's medics and builders. They actively suppress inflammation, promote tissue repair, and encourage healing and cellular regeneration. Under normal, healthy conditions, an effective immune response transitions smoothly from an aggressive M1 attack phase to a calm, restorative M2 repair phase. The soldiers clear the field, and the medics rebuild it. However, in the presence of highly destructive snake venom, this system breaks down completely. The immune system becomes trapped in a hyperactive, endless M1 loop. It just keeps sending in the attackers. It causes the body to inadvertently destroy its own healthy tissue in a misguided crusade against the localized toxin. Pinland's team made a truly remarkable discovery when analyzing the tissue of the treated rabbits. The botulinum toxin fundamentally altered the actual population of macrophages at the site of the bite. The muscle tissue of the toxin-treated rabbits contained significantly fewer aggressive M1 macrophages and a vastly increased number of healing M2 macrophages compared to the venom-only group. The researchers hypothesized that the botulinum toxin effectively acted as a biological switch. It literally toggled the macrophages out of their destructive inflammatory setting and forcefully pushed them into their anti-inflammatory tissue-repairing form. Instead of allowing the immune system to tear the limb apart in a misguided attack mode, the botulinum toxin forced the body to immediately enter a repair mode, entirely shielding the muscle from immune-mediated necrosis. The implications of this discovery are absolutely monumental for the field of emergency medicine and for global health as a whole. David Williams, a renowned herpetologist with the World Health Organization based in Melbourne, Australia, has long emphasized the critical, desperate need for deeper intellectual and fiscal investment in novel, timely snakebite therapeutics. Because botulinum toxin targets the host's own immune response, rather than trying to bind to specific, highly variable venom proteins like traditional antivenoms do, it holds the unprecedented potential to be a universal, broad-spectrum treatment. Think about that for a second. Whether a patient is bitten by an Asian viper, a massive African puff adder, or a North American diamondback rattlesnake, the underlying inflammatory cascade that destroys the human limb is largely exactly the same. A medical treatment capable of reliably shutting down this localized destruction could fundamentally change the standard of care in every hospital worldwide. Furthermore, this incredible breakthrough highlights the immense power and efficiency of pharmacological repurposing. Developing a completely novel drug from scratch is an arduous, agonizing process. It can take decades, it can cost billions of dollars, and it frequently stalls in clinical trials due to unforeseen toxicity or a sudden lack of efficacy. But botulinum toxin already has an extensive, rigorously tested track record in human medicine. We know exactly how it works. It is already mass-produced, deeply well understood in terms of its safety profile and its dosing, and crucially, it is already readily stocked in hospitals and clinics across the globe. If its efficacy as an antivenom adjunct continues to hold up in further rigorous testing, the pathway to deploying it in emergency rooms could be incredibly, unprecedentedly fast. It represents a highly strategic pivot, transforming a compound previously relegated to cosmetic dermatology and neurology into a frontline defense against some of the most neglected tropical diseases on Earth. Now, while experts like Rosetto and Williams eagerly acknowledge the extraordinary promise of these findings, they also carefully caution that the research is still in its early stages. Animal models, while highly informative and crucial to the scientific method, do not always translate perfectly to complex human physiology. Rigorous clinical trials will absolutely be necessary to establish the exact dosing, the precise timing, and the strict safety protocols for human snakebite victims. The dynamics of administering a known paralyzing agent to a patient who may already be suffering from venom-induced neuromuscular blockade must be meticulously, carefully studied to avoid catastrophic complications. However, since botulinum toxin has been used safely in humans for decades, the scientific community has a massive, invaluable head start. Looking ahead, we may very soon witness the dawn of a toxic tag team approach to treating venomous bites. In the near future, the standard protocol for a severe envenomation could involve a brilliant dual-pronged attack. Upon arriving at the clinic, a terrified patient would receive an immediate intravenous infusion of traditional antivenom to neutralize the circulating venom and prevent total systemic organ failure. Simultaneously, in the very same room, they would receive a targeted, localized injection of botulinum toxin directly into the site of the bite. While the antivenom works systemically to save the patient's life, the botulinum toxin would work locally to save their limb. It would calm the raging inflammatory storm, prevent the immense swelling from literally tearing the skin apart, and keep the muscle tissue alive and healthy until the body can fully heal itself. There is a profound, almost poetic irony at the very heart of this incredible research. For millennia, venomous bites have terrorized humanity, weaponizing our own immune systems against us and causing unimaginable suffering and disability. Now, human scientists are learning how to actively disarm these ancient natural weapons by deploying an even deadlier toxin, one derived from a humble microscopic bacterium, to command our own immune system to stand down and heal. It is a brilliant, shining display of modern medical ingenuity. We are utilizing the very mechanisms that make botulinum toxin so incredibly dangerous to execute an emergency override of the human body's inflammatory cascade. As this vital research continues, this deadly poison turned life-saving therapeutic stands as a powerful, beautiful reminder that sometimes the most revolutionary, world-changing cures are already sitting right in our pharmacies, just waiting for us to discover their true potential. Thank you so much for joining us on G Fun Facts Online. We hope this deep dive into the paradoxical world of venom and medicine has left you as fascinated as we are. Keep exploring, keep questioning, and never stop marveling at the incredible resilience and ingenuity of medical science.